Sad news is this is our last one this year, and the next one will be on January 10th. And we have a wonderful lineup for next year as well. So hopefully all of you will join us by Zoom and in person. But today we have truly a very special guest who is in person, and he's going to share his exciting research. But before we go, maybe Jasmine will introduce him.
Hello, everyone. My name is Yasmin Solano. I'm a graduate student in Dr. Philip Kaiser's lab. It is a distinct honor to introduce Dr. Kris Saha, an esteemed scientist whose pioneering work seamlessly bridges the realms of biomedical engineering, gene therapy, and bioethics. Dr. Saha's academic journey is as impressive as his research achievements. He was a postdoctoral fellow at the prestigious Whitehead Institute for Biomedical Research at MIT and a science, technology, and society fellow at Harvard's Kennedy School of Government. Currently, Dr. Saha serves as a professor and the Retinal Research Foundation Catherine and Latmire Murphy Chair at the University of Wisconsin-Madison. Remarkably, he is the only professor at UW-Madison to hold a joint appointment in the departments of biomedical engineering, pediatrics, and bioethics, reflecting his interdisciplinary impact of his work. Dr. Saha has made transformative contributions to advancing non-viral genome editing technologies and regenerative medicine. He has authored nearly 100 scientific publications, holds multiple patents, and has spearheaded several high-profile initiatives, including currently leading a $29 million NIH-funded CRISPR Vision program. His research spans the development of innovative biomaterials for stem cell studies and translating genome editing therapies into clinical applications. Beyond his scientific achievements, Dr. Saha is a leader in the ethical governance of genome editing. He has co-authored seminal articles and serves on prominent oversight committees, including the International Society for Stem Cell Research and the American Society for Gene and Cell Therapy. In 2023, Dr. Saha was invited by the National Academy of Medicine to serve as a panelist for the session titled Emerging Leaders in Health and Medicine. Please join me in welcoming Dr. Kris Saha as he shares today's talk titled Nonviral in Vivo Genome Editing of the Retinal Pigment Epithelium to Treat Rare Inherited Channelopathies. Thank you.
Right. Thank you, Yasmin. That was a very kind introduction, and it's wonderful to be here in California and to see the really impressive center here that spans basic science, translation, and also clinical implementation of really innovative ideas and technologies. And I really hope that genome editing becomes one of them in the near future. And so what you see here is a sheet of retinal pigment epithelial cells from the back of the eye, and that is the target cell that our latest efforts have been focused on. And the rescue of a protein that normally would be not there. And thus in green, this potassium channel KIR 7.1. And so by gene correction of that exact mutation in these patient-derived retinal pigment epithelial cells, we were able to see proper rescue of the not only protein, but channel function and some markers of I think, potency. And the hope here is that we can do this not only for this one example, but for almost any inherited retinal disorder. And so that's a very bold vision. I think that's a bold vision shared here as well. And so I'm excited to learn from the community, what's being done here, and also share what we've been doing. Is it on? Good question. It's funny, these always work when you try them. Well, I'll just use the cursor, I think. Let's see. All right. You know, what's really remarkable here in 2024 is that there are patients that have been treated by CRISPR. And so one of the very famous patients here is Victoria Gray, who I was fortunate to meet in just for a brief moment in London at the International Summit. And you know, she said firsthand how amazing and transformative this type of therapy was, whereas this is an ex vivo gene-edited product now called Casgevy. There's also genomic editing going on in genetically engineered T cells that many patients, thousands of patients have taken. Emily Whitehead is one of these children that was treated with these genetically engineered T cells, and these are being edited at the moment and in several clinical trials. And then the one that is closest to what I'm talking about today is this patient, Michael Kalbauer, who was part of the Brilliance trial from Editas, who described being able to see colors at his cousin's wedding. And so these are meaningful changes. I think many folks in this room understand that. And, you know, I think now we have all of these amazing tools to be able to precisely edit the genome. And I like showing this video because it highlights some of the components that are used and also some of the assumptions that are being made as well when people envision CRISPR gene editing. And so there's three components: the protein in purple, the guide in orange, and then there was a third optional component, a DNA template that flew out of the screen. And so once the protein and the RNA come together, the complex scans the genome, and when it finds a match to the RNA, in this case to the mutation that was matching the RNA, the protein can make cuts on both strands of the DNA. And once the cuts happen, the cell takes over. So if the cell has active DNA repair pathways, it will try to heal this break. And if this template is around for it to be used for homology-directed repair, you can precisely write in the sequence on the template into the spot that you decided to cut. And so this is one of the earliest types of CRISPR, certainly a breakthrough, awarded Nobel Prizes, and really, I think, transformed all of life sciences. What we've been working on collectively in the field is to diversify this type of process, make it more precise, and try to be able to precisely write in a desired sequence into a spot in the genome. And so what I think the SCGE is thinking about, and this is a cartoon form, but it is, I think, in some ways, the workflow of how we envision, hopefully, CRISPR would be used the clinic and have an impact. And so the hope is that as soon as you're able to diagnose a patient and read the variant that you think are causing the patient's symptoms, the physician can order a CRISPR drug that can be administered for treatment. And you know, a lot of this relies on being able to reliably either read the genome, which I think others and the Moore's law type of advances in genomics is already enabling, and many of you are familiar with that. But it also requires kind of building up a set of CRISPR drugs that is safe and efficacious, and that could be manufactured and implemented and given to a clinician in time. And I will say we're not here yet. We're not able to make a CRISPR drug in time at a reasonable cost for a given variant. And so this is kind of a grand challenge that not only I'm part of hoping to solve, I think many in this room are hoping to solve too, but NIH collectively wanted to be able to use CRISPR as a platform. So for instance, if you're able to see this mutation here that we see in one of our patients, this is not my patient, but this is one treated by David Gam at UW-Madison, that at least in best disease, we would be able to intentionally and precisely make that P into a C, right? And so the challenge really here is that it's not easy to develop a drug that's that precise, and certainly was a dream 12 years ago when I started my lab. But as I said, the eye is I think the best place to try to explore some of these development of CRISPR drugs. And so, as you all know, the eye has this beautiful structure where towards the back of the eye, this retinal pigment epithelial layer frequently encodes a number of proteins that are very important for metabolism of all sorts of components in the retina. And channel function, specifically in that layer, can lead to disease. So one of the mutant proteins that I originally described in the first slide, here 7.1, you can see through immunostaining is in this back part of the eye in green, and it's really within the localized towards this area. And the beauty of this, excuse me, is that we can go and directly inject material right next to that layer. And so in many ways, we are taking advantage of advances in ophthalmology such that you can administer safely these types of drugs. And so this is from Luxturna's package insert. And then be able to try to do this genome editing. And so I mentioned that best disease mutation, and if you look across the other types of channels expressed in RPE and also in many other retinal diseases, you'll see a map something like this. And so this is that gene KCNJ13 that encodes that potassium channel. These are usually pretty complex proteins that are multimers. And if you look at the variants that people have kind of mapped here, from kind of the N terminus to the C terminus, you'll see there's certain hotspots that are tolerant of mutation, some that are not, some here that are red that cause Leber's congenital amaurosis. And each of these different sequence variants may require different guide RNA to precisely edit and fix perhaps that mutation. And this is our example that I'll be talking about more. And this, sometimes you can find a strategy that is mutation independent. So you could perhaps correct upstream here at the beginning and bypass any mutant sequences downstream. Those are strategies that could be useful. But in many cases, you want to actually go into the actual pathologic allele and be able to modify it precisely where the mutation is. And so that is a, I think, a harder challenge to develop as an array of CRISPR drugs as a platform. And fortunately, we were able to be part of this genome editing consortium that really started in 2018 throughout the country here. And it really focused on a number of different tool development types of projects. So there was David Liu's group, Charlie Gersbach's group, and others that were developing new types of editors, epigenomic editors, base editors, prime editors. There was our group that was developing new biological systems like human organoids and tissues to test new editors. There was new delivery vehicles that we were also part of in phase 1. And I was fortunate to lead this group and kind of understand that there are really two types of editors that would be very exciting to use in the retina. in particular, and this was a slide really taken from 2021, which in some ways is outdated now, but the two types of editors I'll talk about today is the Cas9, the original one in the cartoon, and then the base editing option that fuses a deaminase to that Cas recognition domain. There are really exciting, I think, opportunities in epigenetic editing, and then directly evolving all sorts of new types of recognition domains in the Cas portion. And what we've done in this consortium is develop this toolkit. So we'd love for more people to use it, and I think it will be populated with more and more data as projects get finished and get published. And we hope that this is a resource such that if you have a cell type and a mutation and a delivery system in mind, you might be able to find some publications that are relevant and some data that sequencing data, imaging data, et cetera, that would be useful for your future project. What happened here in 2023 is that the second phase of this project, this consortium, really wanted to explore this platform idea and take many shots on goal in terms of getting a therapeutic to an IND, an investigational new drug application, and enable first-in-human trials. And so the largest of these awards from this phase 2 was this advanced therapeutic development studies, accelerate the clinical development and evaluation of novel genome editing therapeutics. And so these are kind of team science grants, really complex, large teams that I'm learning how to manage and get a product into an IND. We also have quite a bit of new technologies like long-read sequencing that smaller groups are working on. And this goal here, establish new regulatory pathways in blue, is quite interesting because one of the challenges of taking a formulation to the clinic with one mutant allele or target in mind is that if you have a different target and want to switch out the guide, as I referred to earlier, for, let's say, another mutation that a patient has, right now it would be considered an entirely new product that would require all of the stages of preclinical work, potentially with a small animal study and a large animal study, and then phase 1, phase 2. So, you know, you don't want to have to spend tens to hundreds of millions of dollars to develop a new drug if you think some of the molecular mechanism is in common and you can assess the safety of any changes. And so one of the kind of ideas here that not only our group is exploring, but other large groups targeting the brain, the liver, is that you could potentially cross-reference some of the data on delivery, safety, toxicity in small and large animal studies, and also humans, if you're that far along, to develop another drug targeted against a mutation causing largely kind of the same disease. And so I'll make that hopefully more concrete as I go along, but the idea is that once you can de-risk potentially the delivery in terms of safety and demonstrate perhaps some efficacy, you can start to formulate these drugs in a more diverse way such that you can target many more mutations. A lot of the decision making there and power is at the FDA. And so what we are doing is we are sharing our correspondence with the FDA such that, one, we can internally learn what their concerns are of these different formulations, And two, we're hoping to also share a playbook for the field such they can also learn from what we're doing. And it is in some ways, I think, reflects the philosophy of doing drug development in academia, whereas I think there are many other interests in industrial and corporate sponsored programs that would have barriers to sharing some of that information. All right, so with that introduction in mind, the rest of the time I was going to spend on really two targets, which forms the basis of this CRISPR Vision program that is supported through the Somatic Cell Genome Editing Consortium. And they are these channels. One is Tier 7.1 that I introduced earlier. There? Oh, no. Okay. So it's a potassium current, and mutations in this gene are recessive. So you usually don't have any of the protein. And the goal here is to restore proper expression and form this channel and enable current, specifically the potassium current, to get through in these cells. The second target here is a autosomal dominant disease, thus disease. And this is a calcium activated chloride channel. So chloride goes through here in the yellow balls. And there, the goal here is to destroy the mutant allele because in some of those mutations, it disrupts the channel function, either by disrupting the assembly of the full channel or in other ways that we don't fully understand. But we've been able to show that if you're able to destroy and produce indels in the mutant allele, we can rescue function by just expression from the non-mutant allele, the wild type allele. And so our team actually is pretty diverse. The bulk of the work has been done here at University of Wisconsin in Madison. We have John Watts at the University of Massachusetts Medical School, who's been internally making chemical modifications to the guide. And one of our projects has been led by Spotlight Therapeutics, a company that has made engineered ribonuclear protein. So, go to the next slide. This first example here, the Cure 7.1, again, we're trying to correct this mutation. And our strategy here is to encapsulate a mRNA in a lipid nanoparticle and deliver it next to the retinal epithelial cells through similar strategies as used in Luxturna. Some of the jargon here is the ABE8 is a base editor that targets A bases that has been evolved to be highly active, and a guide RNA that targets specifically the mutation thus causing the disease. And so a little bit of introduction to Leber's congenital amaurosis. It's a rare disease. It has these clinical features, and it's about 5% of retinopathy. There's many genes that have been identified, and I showed you some of the various mutations within a gene in that crystal structure earlier. So the patient that we've been working on the most has been this patient that has this single base change from a G to an A. So that's in the gray box in the middle here. And therefore, A base editing could revert this premature stop codon, the TAG, to a TGG, and then restore full-length protein production. And so we have blood samples from the patient, some of his parents and siblings. And so this allows us to reprogram them, make induced pluripotent stem cells, and get a lot of genomic DNA and also cells to do some of the work. We've also spent the time to make a mouse model that I'll be going through. So when we started this work, we thought, okay, Luxturna is working well. There's AAVs and a lot of tools out there to deliver to the RPE. I think there's been some appreciation of the challenges of using that approach, although I do think it's still quite clinically actionable and of course relevant. We've been trying to avoid some of these challenges in making viruses, and been developing a series of nanoparticles that have various different chemistries. And so I don't have time to go through all of them, and really all of them were done in collaboration with Sarah Gong, who's next door to me at UW-Madison. And one of the things I want to highlight is that many of them fall apart here inside the cytoplasm through this glutathione responsive release. And so the disulfide bonds really go away inside the cell once it's in there. And then to facilitate this escape, I'm not sure why it's doing that, in the middle here, the endosomal lysosomal escape, some imidazole groups in the kind of structure of these nanoparticles helps generate a pH effect. So in combination with various types of chemistries, we're able to load not only mRNA, but also ribonuclear proteins, as well as plasmid DNA. And so it's been pretty versatile. The original kind of idea of putting imidazole and disulfide groups was to make what some people called, after we made it, a shrink-wrapped ribonuclear protein. And what we do is we essentially have charged monomers that coat the surface of the protein RNA complex, and then we're able to do some polymerization in situ to make a cross-linked shell around the RNP. And so it really did matter to get all of the monomer compositions right. And so this is published. I won't go through it extensively, but on the y-axis is a reporter cell line where you'll see the higher the bar is, the more editing you have. And getting the ratios of all of these components right really did help. And at the end of this type of a process, when we look at where the protein is, the Cas9 protein is within a single cell, so this is one representative cell, we've tagged the endosome in green, you see that there's quite a bit of our payload protein inside the endosome by the yellow on the far right. But there's certainly some of these red and kind of purple splotches on the far right that indicate that it's inside the nucleus, at least at this time point. And it's really only a matter of a few hours that you see some of this signal. If we look under microscopy, these are fairly small particles because in some ways they're only kind of a thin nanometer shell. And what we can do is we can freeze dry them and they retain activity. So this is exciting in that we're able to make shelf stable type of product. So the last thing that we kind of played with was what could we attach on the surface to help uptake. In this case, this is a cell penetrating peptide, and the one in green here has the cell penetrating peptide. And you'll see that we are doing better than when we don't include the cell penetrating peptide, which is in purple. So the y-axis here is the amount of editing we see in vitro. The viruses, of course, do very well. So we're not reaching viral delivery levels, but we are able to bias kind of uptake and editing. And one of the tools that we started using, this was back in 2016, 17, is this AI-14 mouse, which is from the Allen Institute. And it was originally engineered such that Kris would excise a stop cassette and turn on TD Tomato. I know there's been some beautiful strains here, and this it's probably an antecedent to that, and not as robust a reporter, but still it's a gain of signal, and we think gain of signal is quite nice in terms of seeing whether a cell is edited. And so what we ended up doing was conjugating all-trans retinoic acid because we knew that this molecule can be bound to IRBP and preferentially transported to RPE as part of the visual cycle. And so we put those nanoparticles into these mice, injected it into the back of the eye next to the RPE, and then did the flat mount microscopy to see where TD Tomato was. And so when we had here one particle that was injected through subretinal injection, you can see some splotches of red in the middle at this dose. And when we conjugate that all-trans retinoic acid at the bottom, you can see kind of a larger area of the floret that's been edited. And what we see here is on the order of 2 to 3-fold enhancement of the area that's been edited by adding that all-trans retinoic acid. And so these are ways in which we can bias uptake towards a particular cell type. We also developed these other particles that have a different internal structure, but again have the disulfide linkage and the imidazole. In this case, we loaded it with mRNA rather than proteins. And we weren't able to make that shrink wrap around mRNA because it is not as solid a core to do our chemistry. So we use this structure, and this also can be generated. We get particles that are a bit larger in size, between about 20 and 100 nanometers in diameter. And here we tested it in vivo, in this case, against a mouse that was now available for us to edit. This is a same gain of a stop codon, the W53X mutation that we see in patients, but this is in the endogenous mouse KCNJ locus. And we had to design a slightly different guide due to some small differences outside of that mutation from human to mouse species difference. But we have a good guide for A base editor. And so what we did is we looked in a model where we have one allele of the A base editor, and we inject with these particles that have either the guide against a portion of the genome that's, or it's a guide that's non-targeting, meaning it has no match to the mouse genome, or we have a base editor with a guide targeted to that W53X mutation, as I showed in the previous slide. And what you see here is this multifocal ERG taken from these mice treated with these particles. And essentially, you see pretty flat responses when we treat with the mutant, the non-targeting guide. And then in some cases, we get in green some localized areas of really high response. And so on average, if we look over time as well, we see a pretty durable response. It's significant, and we think we can do better if we increase the dose and move to our lipid nanoparticles, which is our newest generation that we're working on at the moment. And so we're excited by these results. It's actually it's been published now We are testing this in human-based models as well to see whether this strategy is appropriate for large animal testing. So some of the studies here that we've done, I've mentioned the induced pluripotent stem cell based differentiation, and this is in collaboration with David Gam, who makes these cell layers. We see in our newest version this rescue of the protein, whereas without treatment, you see no green in the middle, and on the right, you see green and also quite a bit of localization to the membrane. We've also been working on doing automated patch clamping of these edited cells. And we essentially see a flat line when we do these single cell traces and adding rubidium and potassium, and specifically looking at the potassium current flow. And our treated ones have notable conductance. Some of them don't have full proper conductance. So we're trying to understand what genomic outcomes there are, and maybe perhaps some effects from the LNP. And so these are pretty early data, and we're collecting more and more of this. And so in terms of looking at on and off target precision, what we see among our formulations here is that we have several, 6 of them that get on the order of 30% gene correction in these human iPS cell systems. Some of them don't do as well here, but certainly we're looking quite a bit through additional sequencing technologies using long read, and also looking at off-target sites. And so some of that work is also enabled through this consortium. There's a new study from Chandra Sai's lab, who originally developed GuideSeq, which is this way to look throughout the genome in a test tube of where Cas9 would cut. And he's adapted it now to base editors, so he can map where base editors would make a modification. I won't go through the details here, it's in the preprint, but when we did this with our guide design, we saw a very high specificity, probably the highest I've seen for A base editing, which had a specificity of about 0.9 out of a maximum of 1. And in some ways, it's due to the sequence context that there's no A's nearby within the base editing window. And so we're fortunate in that way in this target, but in other targets, we may not be so fortunate. Regardless, we've taken the other sites that are nominated through this assay. This is in vitro assay, and we've looked now in our treated any types of modifications there. We really don't see many, or I think only in one case, the one that's boxed, do we see a modification above our background level of error in these assays. So we think these transient strategies of only expressing the editor have very high precision. And we are trying to leverage that in our second part of our program, which is focused on BEST1. And so, as I mentioned, this is a dominant mutation, and therefore we need to actually have very good precision. The top off-target site, when you look at a particular mutation, is usually the green allele. It's the wild type because it is only differs by 1 base. And in this channel, there's been a number of different mutations that have been mapped. Some are in the so-called class region and the chloride binding region, and then some in structural regions. So the ones that we've been looking at are A, 146K, so alanine in the 146 amino acid position, N296H, and then also R218C. And here we're taking advantage, again, of our collaboration with Dave Gam to make not only retinal pigment epithelial cells, but also some retinal organoids. And we think the organoids are important potential off-target tissue that we want to ensure functions properly. And then we could also do the on-off target studies in our iPSC RPE. And so this study here took these iPS cells from 4 different patients, reprogrammed them, and then differentiated them into these RPE layers. And to summarize a lot of single cell patch clamping, the wild type has a nice current density that's in green, that's kind of at a 45-degree angle when you look at this patch clamp trace. Patient 1, 2, 3, and 4 really have flat, no calcium activated chloride activity. And we also spent the time to correct this mutation in one patient, this patient 4, and look at its single cell activity. And what we see is when we correct it at the iPS cell stage and differentiate it, we get full rescue of the calcium activated chloride current. And so one example of this is to do this specifically in the RPE, not at the iPS cell stage. And we designed this guide that has specificity in the seed region of the guide to the mutation, the C to T mutation that I showed you earlier. And this is an exon 6 of this BEST1 gene that encodes that channel. So what we see here in iPSC RPE that normally you have a 50-50 mix of your wild type and mutant reads. So this percentage in red really should be 50%. And that's what we start off with. But after gene editing, we get that read go down to 12%. And the rest of the reads end up having indels right around the predicted cut site. And what's surprising to us is that even though there's a lot of different outcomes here, about 95% of these outcomes are frame shifts, which trigger premature stop codons and nonsense mediated decay of the transcript. And so this set of outcomes is actually different than what we saw in iPS cells, which are cycling, where we had less frame shifts. So it indicated to us that actually the repair outcomes could be dependent on the cell type you're looking at. In this case, that's great for us because we want to destroy that protein, and these frame shifts will do that. And so we went on and looked at the various different patients, and we saw similar profiles. And so that work is ongoing as part of that U19 project. I wanted to actually mention this project that is looking at whether we can see the effects of these types of genome editing treatments on human photoreceptors. And so through stem cell differentiation, we can also not only make that sheet of epithelia, but we can make these optic vesicles that essentially what many in the lab call golden Cheerios. They float inside the dish, and they're kind of an inside-out orientation of the inner retina. And so what we ended up doing was trying to deliver our genome editors to these organoids, and we just had trouble transducing the photoreceptors, which are actually not on the outer rim fully, but the nuclei are a little bit inside the surface. And so some of our students decided to dissociate co-incubate with some of these viruses and then plate, certainly it destroys the beautiful structures that are made, but we are able to keep the photoreceptors alive for several days to weeks. And our goal here was to look at transcriptomic perturbation specifically in photoreceptors. And so this is in some ways the worst case of when you're trying to target the RPE, is that you mess up the photoreceptor. So we wanted to see what would happen if we have a lentivirus, in this case as our delivery vector, targeted against a master regulator of photoreceptor development, such as NRL or CRX. The other thing that we can do is before we dissociate, we can do some 2-photon imaging. And so this was done by Melissa Scala, a scientist in her lab, with these same organoids that were treated with the genome editors. And he was able to take these videos following this chemical treatment of retinal, and look specifically at the outer segment. So I'll play it again. It is a little subtle to see where and when these light up. And so he looked at this response, essentially simulating phototransduction to find a specific signal here in 2-photon microscopy of this retinal at about 45 minutes. And so there is a publication on here that isn't showing here, but it is on our CRISPR Vision website that you can find, that has the phasor diagrams and the details of how we can get a functional response of these organoid-derived photoreceptors using this treatment. And we also have used this lentiviral delivery vector, as I mentioned, to deliver a guide plus, in this case, Cas9. And when we do our single-cell transcriptomics, we're able to track the cells that have the editor. And what we see here is that we can edit about 30% of the photoreceptors. And we do lose, in terms of some of our entire data set, the total number of photoreceptors that we've profiled. But we still have, I think, a pretty good sample set of about 10,000 cells that we can identify using clustering as photoreceptors. And we've been mapping out which gene regulatory networks are being turned on and off when we either cut a master regulator like NRL or CRX, or have a non-targeting guide. And so we're hoping to develop a set of biomarkers that we can assay, not through single-cell transcriptomics, but through standard qPCR that could be implemented in some of the cGMP spaces, or in preclinical work that's much easier. Okay, so I'm gonna end up here so we can have some time for questions. We are looking at doing as much as we can with these human-based models, as you can see in Best Disease and LCA. We think it's a really robust platform for looking at the precision at the DNA level, at the protein level, and at potency level. And this would enable and make that platform such that we can provide a data package to the regulators that, given a new mutation, we have data showing that it would be safe at these types of levels in a relevant cell type. And we're hopeful that this approach could be used for other mutations that are affecting the retina. Many of them are here in this review, and using other delivery strategies to edit photoreceptors and other cell types that are affected that lead to blindness. So overall, I would say that our strategy thus far has been to use a non-viral approach that we think can be customizable and ideal for rare disease. And we have these capabilities that have been supported through our SCGE funding generously to build up this infrastructure to explore whether CRISPR could be a platform. These are the folks that did the work. I want to especially highlight Sarah Gong, who did all of the formulation work and developed the chemistry. Bikash Patnaik, who recruited the patients and has a long-term focus on Cure 7.1. David Gam, who we worked collaboratively on the best one and is a pioneer in making these retinal cell types. And then in terms of lab members, Amr led the nanomedicine program and development with the Gong Lab. Divya, Kayvon, Kawan are all very active members in our U19 team. And so it's really a large effort, and happy to share and answer questions.
Chris, thank you very much. We have a few questions, I guess, from Zoom.
All right. So the first question goes to Samantha. Samantha, are you ready?
Sure. How big would you say your shrink wrap particles are?
Yeah, they're about 12 to 20 nanometers. So that's based on dynamic light scattering as well as some of these DM measurements.
Okay, thank you.
Yeah, that's in the Nature Nanoscience paper, Canadell.
And Tietle Pansi, are you ready to ask your question?
Yes. How long were these organoids differentiated for?
Yeah, so that actually is a great question because we didn't know how long to go, and the shorter would be actually easier. We have data on 75 days, 100 days, 165 days, and 200 days. And so we looked at all of those, either 3D and 2D, and we tried to figure out when is the earliest time where we could argue to someone that is photoreceptor-like or enough. And actually, that was not a very easy question. We essentially divided up our data set into early, mid, and late. And we think the earliest time point where you can get cells that at least express some of the core transcription factors are on the order of 75 days. But of course, as you go further, you get more rods and cones. And so there are benefits to waiting longer. It really depends on, I think, the type of adverse event you want to detect and expect based on your editor. So in a viral preparation, it might be that you need to go longer for some types of perturbations. And if it's something that is a genomic off target that you know a particular gene that is expressed and you see that expression early enough and can monitor dysfunction of it, you might be able to get 5 earlier. So it's a long and complicated question, but it is actually a difficult question that we've been trying to answer with our data set.
And in follow-up, Leila Bahmani, can you ask your question?
Yes, very nice presentation. Thank you. I have a question. You mentioned some downside of viral delivery, but I'm curious if you think there is an important side event for the nanoparticle delivery.
Sorry, I really had trouble understanding. It was a bit choppy. Did anyone gather the question? Did you ask it again, maybe more slowly?
Oh yeah, can you hear me?
That seems better, yes.
Okay, so I have a question. So you mentioned some downside of using the viral delivery, but my question is that do you think is there any downside for the nanoparticle related delivery as well? What was the most problematic difficulty that you had when you used the nanoparticle delivery?
Yes, great question. Yeah, I think overall it's the lower efficiency. I think in our case, we are able, we argue we should be able to see a therapeutic response at about 15% or so editing of the entire RPE layer. And we don't need 100% or 80%. And that's why we think it's a good target to go after. But certainly if you do need a cell type that needs high efficiency editing, you would likely go to a viral vector. As well as some cell types that have really nice tropism for viruses. So photoreceptors in our case are pretty hard to target with our nanoparticles. I think it's a very dense layer that the particles have to get through and to. And there are some AAVs that can get there. And so in those cases, I do think at the moment, viruses are probably the best way to go.
Hi, Chris. This is great work, and thank you for sharing these exciting studies. So when you were talking about mRNA versus protein delivery using nanoparticles, have you seen the difference in efficacy, like whether the protein delivery was better versus mRNA in vivo? I'm talking. I think you showed some in vitro data.
I'm trying to think of the right study. We haven't seen in vivo a big effect. We actually haven't looked at that very carefully with the same guide and the same target. I think in vitro, we've seen against some targets, not specifically in RPE, but longer expression and protein activity through the mRNA strategy, and therefore higher on-target and potentially off-target editing. And so one of the aims of our BEST program is to do exactly what you propose, look at mRNA versus protein as our payload. And right now, our hypothesis is that the protein delivery should have less off-target effects on the wild type, because the activity is still limited to its turnover inside the cell. We haven't looked as closely as we want to, but we're planning to do a study.
Thank you. So second question I had is, you mentioned the nanoparticles you made are like a smaller size, 10 nanometer. But the ones we make are a little bigger. So have you explored if you just give an eye drop, if they penetrate the cornea or something?
We haven't looked at the eye drop, but it's a very interesting question. We have done some work with intravitreal injection.
Yeah.
We were hoping that it would get to the photoreceptor layer. We saw some signal there in our PD tomato type assays, but we didn't see as robust as we really wanted or saw it through subretinal injection for the RPE. I think there's quite a bit of chemistry and formulation work that could be done that could potentially get it into the cell type we want it to get into. But yeah, I think the challenges through intravitreal and also, through drops are things that could be tackled in future work.
Chris, really nice talk. So I have a question. What you show for the best one, right? Editing is very high efficiency. So I'm wondering how that in vitro data can be translated in vivo. Also, do you see the efficiency can be translated from RPE to retinal organoid, right? If you try that, do you see the same level of efficiency and specificity?
We don't see the same level of efficiency as the lentiviral. So I think on average, we had about 60 to 70% depletion of the mutant allele with virus. That's about 2-week constitutive expression of Cas9 and the guide. With our transient ones, we are in the 10%-ish range at the moment with some of our formulations. I think we actually can do quite a bit better. Some of the challenges has been the right dosing in vitro that correlates to what's in vivo, and actually some of the media supplements and health of the RPE that would mimic what would happen in vivo. And so those, I'm not giving a full answer, but those are challenges that we're trying to explore, and we welcome some ideas on how to make our in vitro system more correlated and predictive to what we see in vivo.
Just another deep, small question. I was just curious. So when I look at the dissociation of the organoid, right? You do the profile, you look at different cell type. It seems like the percentage add up is way less than 100%. So it's like one wondering what happened to the rest of them.
Yeah, it's a good question. Let me see. There are certainly cell types that we can't easily cluster cleanly. That might, and so our challenge through the answer to the first question was, is that because of genome editing in some of our samples, or is it because of different or incomplete differentiation? And in some cases, we can't answer that, but in other data sets, we have the paired untransduced, no editor side by side. And so I actually think part of it is really not having very clean transcriptional markers for the developing cell types. And certainly not every cell matures at the same rate too, right? So that's usually, I can easily pull it up, but that's usually the reason I didn't put maybe, we just call them other instead of photoreceptor, yeah.
Hi, thank you for the great talk. You mentioned that the organoids don't respond very well to virus transduction, and I'm wondering if you tried manipulating them through electroporation.
We haven't tried electroporation in that project, mostly because I think we had viruses that we were able to get working by dissociation. And I've heard, although we haven't done these experiments, is that these photoreceptors are pretty sensitive to the stress of electroporation in 3D. So they're precious samples, especially after 200 days of differentiation. So those are the kind of the reasons practically why we haven't explored it, but it could work.
Very nice talk. I have a question about the decoration of the nanoparticle. It seems like the delivery efficiency increased a lot by conjugating the R-trans retinal on the surface of the nanoparticle, but it seems that this is really cell type specific. So I'm wondering if it's possible to like design different decorations for different cell type, or kind of discover a universal decoration for like most of the cell types. Is that the future direction on the nanoparticle delivery system? Thank you.
Yes, short answer is yes. I mean, I actually think some of the work with exploiting natural pathways of the visual cycle and other physiological changes in either a healthy or diseased patient is a very exciting way to go. There's been a lot of work targeting the liver, and it seems like using ApoE and the cholesterol kind of natural transport pathways to promote uptake in the liver has been the most successful there. There's certainly really impressive work out in the field that have conjugated all sorts of peptides and targeting ligands and libraries and injected them in vivo to try to see where they go. I think in general, they've found that this ionizable lipid in the LNP is a stronger determinant of cell type specificity or tissue type specificity than the peptides or other molecules that you can conjugate on. But it's really dependent on size and density of all of these molecules and which systems you're looking at, route of administration as well. So we don't really have to worry about the bloodstream here, right, through subretinal injection. If you're trying to do systemic targeting the liver, you really do need to worry about what's going to coat your particles.
So, Kris, you mentioned about decorating those particles with retinoic acid, and so it's reduced on it. Have you proven that it binds to IRBP? Because I bet it doesn't. And what you have is only hydrophobic tag that enable partition to membranes. Now, the binding site in IRBP is pretty deep, and so there's no way you could.
Okay. Yeah, I mean, it's a good point. We haven't looked specifically at IRBP. Have the data that we see editing outcomes, but not specifically binding with that molecule. Okay.
Hi, great talk. So my question is slide 43, where you showed like 4 patients.
Mm-hmm.
And like first to have visual, like was it done in the patient eye, or was it like the cells taking from the patient eye and then like edited outside? And my second question is about after doing the edit, I see the current density in the edited eye is higher in comparison to the wild type.
Yeah, those are good points you bring up. Certainly, I skipped over the details. Let me just bring up that slide. I think it's this one. So this is certainly not biopsies from patients. I didn't mean to represent it that way. It is blood samples taken from patients, reprogrammed to pluripotent stem cells, and then matured into these RPE layers. And so that's what these images are. This is staining for the protein. This is a recessive form, so you see arguably background levels of the protein. And at the bottom are representative single cell voltage current traces. And so I think your question was, once we did the correction at the iPS cell stage, that we see a little bit different slope, or at least this corner is a bit different. I think that over all of the different outcomes didn't end up being a significant difference over wild type, but I'd have to go back to the paper and look at that. Certainly there is a potential that we have a different current upon editing in this scenario, because we don't correct the mutation, and we have a spectrum of alleles that are there. So products are actually messing up with some of those channels. We don't have the resolution to see all of those outcomes at the moment. Perhaps we will in the future, but overall, we do think that of the cells that we were able to profile in that manner, we had similar outcomes to wild type on average.
Hi, Dr. Saha. A great talk. I see it as a foundation to enable future CRISPR therapies. I have a question. I see your focus on Cas9 nuclease and editing base editors. Do you have any insight how translatable is the prime editor, if there is any evidence of feasibility of using in therapy? Thank you.
Sure. Yeah, I'm very excited about prime editors as well. I think the major challenge in the field has been to make high quality guides that are very long, because you have the template that needs to be reverse transcribed. I think there's been a lot of advances there. And so that I don't think will be, a long-term bottleneck. And so I think an open question is how do these strategies, kind of older generations of CRISPR arguably, would coexist with a new potentially disruptive prime editing strategy that could do these same types of edits. At one level, I think you can always go for like the Mercedes-Benz version, but if the Toyota Camry can do it, it might be good enough. So that's the short answer, but I do think certainly for mutations that are deletions, where you need to write in something, relying on homology-directed repair is probably not feasible in many cell types, especially in post-mitotic cells, and prime editing is the way to go.
Hi, my name is Hewitt. So my question is regarding BEST1, when the goal is to destroy the genomic mutant in conjunction with gene augmentation, have you considered targeting cis-regulatory elements as opposed to specific mutations and relying on the NMD pathway? Or would that create some sort of greater complexity in terms of that?
Yeah, so for the dominant case, usually the cis-regulatory elements aren't mutated. You have the same alleles that are driving transcription. But in Casgevy, the cis-regulatory elements are super important, right? Because that was a way to get an indel in a repressive region that could turn off fetal hemoglobin. So certainly, I think there's a lot of promise in editing cis-regulatory elements. In this case, I don't think there is one that we could target.
Hi, Kris. Thank you for sharing. Thank you for sharing one of your insights. My question is kind of like, have you ever monitored the long-term post-delivery effect of the ion channel? Like you may know that nanoparticle formulations may not provide like long-term expression or effect, which may lead to the repeated dosing requirement for effective therapy. I think my question is kind of related to like how stable that such delivery platform is in mice.
Yes, because we're making genomic changes, I think the effects are durable. And that's one of the major reasons to go to genome editing, that we don't hopefully have to redose. That said, if we're not able to reach the efficacy with a single dose, there is an opportunity to redose with a non-viral formulation in particular, because the hope is that we don't have the same extent of viral or immune response to the vector as you would do with a viral strategy. So in kind of the liver targeting programs in vivo that are non-viral, they are starting to redose, I think, and so far haven't seen any adverse events from that. And you then get another shot to increase and potentially maybe double your efficiency in therapeutic effect.
Hi, Dr. Saha. Thank you so much for such an interesting talk. So you mentioned about the editing strategies that you use for example, BEST1, and I'm piggybacking off of Rafal's question about prime editing. But have you ever investigated strategies such as exon replacement, where you can achieve what you mentioned of fast tracking disease that have multiple mutations and that share similar mechanisms?
We haven't, and we'd love to. I mean, I think it helps us. Certainly there are hot spots if we go back here around these different domains, calcium binding class, that could have an exon replacement potentially by prime editing, and that might hit, maybe 30% of the patients or 50% of the patients rather than, single-digit percentages.
Hi, Kris. That was really nice, and it's beautiful to see how the consortium is working and help each other. I had a question again to specificity. So I wonder how specific actually those retinoic acid labeled particles are. I understand they are more efficiently labeling, I mean, targeting RPE, but did you try to see how, whether they do actually are specific to RPE? I did not caught that if you mentioned.
We did also look at the neural retina from the same animals, and we did see some sprinkling of red essentially. So it's not 100% specific, but certainly we saw greater uptake.
So so my second question is actually to think opposite way. So if you can target something to photoreceptors based on the physiology of the cell, have you tried to look at the maybe, for example, very long polyunsaturated fatty acids, nanoparticles, or some LDL, HDL composition, so to mimic it in the nanoparticles? Did you think about it?
I've thought about it. I haven't done it, and I'm very interested.
But the cellular are critical because and they're endogenous promoter, so it doesn't really matter if you correct in the cell. That doesn't need to be corrected.
Right. I think there's now, especially with mRNA formulations and some of the viral formulations, 5' UTRs, as well as the promoters used, can give you the cell type specificity. It's kind of a question of also where do you want the material to go to get the most therapeutic effect. So I do think it helps, even if it's incremental, to get some enrichment in the cell type. But if the major concern is off-target toxicity, using those enhancer promoter elements, if you have space in your viral genome to do that, that's great. In the mRNAs, also, you know, these regulatory elements and microRNAs also can help you tune down expression in an undesired cell.
I have two questions. Fantastic talk, thank you. I was wondering, so I noticed that for the base editors, you tried mRNA delivery. Did you ever try RNPs?
We have those and have not fully looked at them. I think really, maybe you guys have the tricks that we don't, but it's been harder to make some of the base editor proteins than mRNAs. So those are newer capabilities that we're interested in some ways similar to the question for Best disease, could we get better specificity using the protein is one goal, or just higher efficiency too.
Mm-hmm. And my second question would be, could you comment a little bit on the strategies of translating these tools into clinic, and what would be the role of academia versus industry?
Yeah, I like that question quite a bit. I think about it a lot. So academia versus industry and moving these actually to product approval. I think there's challenges for both. One, I would just say in academia, your skill set is usually more open curiosity, less milestone-driven work, and focus required for systematic product development. Usually is not there at many institutions. Of course, there's exceptions and some do it very well. There are cases, and especially in our case here, where the markets are small for a rare disease, that I think academia has a more important role to play, especially in product development. Whether they can get it all the way to the finish line and manufacture products reliably and safely, I think is a tall order, but doable with further investment. So that's kind of the commentary on academia. Of course, industry has taken up, in some cases, rare disease, like Editas in the Brilliance trial. I do think there needs to be quite a bit of open-mindedness to failure and learning from failures and what you define as a failure that may not need to be through the primary lens of return to shareholder, which of course they need to focus on. So it's an interesting, I think, ecology of approaches. I think we're fortunate that both have quite a bit of interest. And you know, one of the more interesting places for the consortium is how we can learn from developments in both sectors.
All right, last, the great conversations, and now I give you Vice Chairman.
Hi, Kris. Maybe that's an appropriate question to end a beautiful discussion, but I know you've also been involved in the discussion of the ethics of this technology, and this will become probably more and more relevant as this methodology becomes mainstream. So can you give us your take on the main concerns and how they could be overcome as this technology becomes, you know, translated to the clinic?
Yes, yes. I could pick up a lot of time on this, but I think it shifted quite a bit. I mean, certainly there was the initial, you know, 2015 summits around germline gene editing and, you know, trying to establish limits on where these types of approaches should go. That I think is in some ways still being developed. There's an international framework trying to be developed, you know, some high profile controversies, et cetera. You know, here we are really focused on somatic cells, and I still think important questions about access are there. And also kind of right to try type of arguments that if a patient really does want to take a risk in a very rare condition, how do we either empower the clinician to say yes, or you know, have some oversight to put in some guardrails. And that's really, I think, a complex question, especially in a healthcare system that's unequal and also a global market where you know, you could potentially manufacture things cheaper in a less regulated country or a place where it's just cheaper to produce certain types of drugs. And is that where innovation should go? So these are all, I think, in some ways, both questions about ethics at a professional society level, individual researcher level, but also larger questions about public health that aren't easy to disentangle. So that is the short answer. Happy to talk more. Thank you so much.